speak very loud after 5 or 10 minutes, so feel free to tell me from the back of the room if I don't speak loud enough. Great. So so welcome to a pan-genomic introduction on construction and visualization of pan-genome graphs. We'll see a few different graphs, and the lecture will be somewhat interactive in the sense that I will be running the tools on small examples. Feel free to interrupt me at any time, raise your hand, ask a question, and I'm happy to be interrupted. A brief presentation of my group at University Pasteur. We are a computer science group working on many bioinformatics topics. Some people are, just one person is here today, Francisco, who is an Alpaca PhD student. Johan is also involved in Alpaca and is not here today. Luca was still in this picture when the picture was taken, but is no longer in the group. We research genome assembly, metagenome assembly, data structures on chemers, searching in large data sets, and today we will talk about pan-genomics. And the mascot of the group is an axolotl. Okay, let's start with the pan-genome intro. As mandatory, I have to give you my definition, personal one of pan-genomics, like every speaker did so far. In my perspective, the pan-genome is a collection of different genomes, aligned or not, from related species, and typically at some point of the analysis task, there is a condensed representation of those genomes, and it's often a graph. Although technically, we could argue that multiple sequence alignment is also a pan-genomic representation, although I never see this type of definition being used in practice. And then, of course, there are downstream applications. Sam has been talking about some of those, and also Yoni this morning, where comparative genomics takes place in finding SNPs, structural variations, presence absence of genes, and so on. I would argue that there are two types of pan-genomics, and we are on the left side during most of this call. The sequence resolution, where you consider all the bases inside genomes. However, for instance, in my institute, many biological and bioinformatics groups are considering the right side. So where for them the pan-genome is a bag of genes, and they are interested in presence or absence or different copy counts in genes inside the pan-genome, but really the gene level. So SNPs are not important, structural variations are not important for people studying bacteria in this framework. Sam, feel free to chime in if you want to.
Yeah.
Okay, so I wasn't so sure, but I think this is a distinction, and converting biologists to the left representation is still an uphill battle. At least at Pasteur, they don't know so well the methods, they believe much more on the right side. So I guess it's our job to do the tools and show the applicativity of those tools so that people do sequence pan-genomics. I will not go too long on those slides because this is the typical applications of pan-genomes I speak about. So this giraffe, just a highlight from last year in terms of genotyping variations. Tomorrow you will most likely hear about ODGI from Eric, which is a framework for constructing and also representing base level variations in pan-genomes using variation graphs. This was human, and so on the bacterial side, I like to highlight two applications. One is from Sam group on the right, RAM tools. You heard it, I think, yesterday, for SNP genotyping and also other things. And on the left side of the slide, I highlight, which is not technically a pan-genome paper, it's more of a metagenome paper, but inside this paper, there is a pan-genome graph. What they did was, what we did, and I'm core for this work, we took several strains of the same bacterial species, constructed a pan-genome graph in the form of an assembly graph of all these strains, and the goal was to separate those strains. So then the graph can be visualized like here, it was the one graph, and then you could construct a matrix with abundance of each unity in each sample, and then from this matrix try to decompose it so that you can actually separate nodes of this bengenome graph into its individual strings, so individual graphs. I won't go too much into details, but that's one potential application of bengenome graphs applied to make a genome assembly. So these are typically the applications I highlight when I give a talk at Pasteur about bengenomics. If you know about other major types of application that I'm not talking about, but I should, tell me after or even now. I'm happy to hear about other potential things. Oh, okay. Yeah, I guess it was exhaustive. So today, in this lecture, I want to present some various types of bengenome graphs, some you've already seen, and especially show you in real time how they can be constructed in practice and visualized. Why? It's because often bengenome graph construction is the first step of any of the analyses you've seen in the previous talks, in the forthcoming talks in this course. And especially there are many types of graphs which you can use. The question, which is the natural one, is which one is most adapted to a particular application. So we'll see what those types are, what the differences are between those bengenome graphs, and discuss a little bit in which case it can be applied and in which case it cannot. The particularity of this lecture is that I will show you all these examples on just two genomes from E. coli species. One is the most famous E. coli type, which is K12. Another one is what is called the Sakai strain, 01577H7. And the particularity of these two E. coli is that they are pretty different. So here I'm showing you a synthetic visualization from a nice paper from Volume 1, 2018, where they plot the K12 genome on the bottom and 0157 on top. You notice that they are different lengths. K12 is roughly 4.5 megabases, and the other strain is 5.4 megabases. And especially, what I find interesting between those two is a large insertion of more than a megabase between those two strains. So the immediate question is, will the bengenome graph reflect this insertion or not? And of course, there are other differences between those two genomes, so we'll see how the bengenome graphs will actually capture those differences. But the takeaway of that slide is only two genomes and very different in variants, big events, and size. The guiding principle of the four examples I will show you is how can one get quickly from genomes to bengenome graph. For this input, from user perspective, are the tools usable, and we'll see how the graphs differ. Right. All the commands that I will type today are on GitHub. And in fact, the slides that you're looking at right now are already on my webpage. You can access them later, so that you can, in fact, find also this GitHub accounts right. So part one, the point class. Before I start, so I have a bias because I have studied the Bruin class for a decade now. I'm not so sure who is super familiar with them, or if it's the first time you hear about them. Who knows the definition of what the Bruin graph is? Like 90%, maybe 80% or so. Who has constructed one? Okay, 80%. Yes. Right. So today you will see one constructed in real time, but it's almost as good as the community yourself. So it's a type of bengenome graph, but it's also coming from a genome assembly. Its definition is that the nodes are k-mers and edges are k-mers per exact overlaps. In terms of bengenomics, it's widely used these days for bacterial bengenomics. So this is two software called BICIR and GWAS, which use the Bruin graphs to perform GWAS, but not the same type as human GWAS, but instead on bacterial species. So genome-wide association studies to find not necessarily SNPs, but k-mers which are associated to some phenotype. And in that case, they look at unity. It's not really important here. So the general principle for those of you who didn't raise their hand so far is that you start from reads or reference genome. In the pan-genome case, you can start from a reference. Extract kernels and then just construct it. The nice aspect of the Bruin graphs is that you can visualize SNPs in a certain topological way, but there are bubbles in the vocabulary of the community. Typically 2 paths starting and ending at the same nodes, but taking 2 different nodes, 2 different setups. To summarize a little bit the advantages and disadvantages of the Bruin graphs for pan-genomics. They are efficient to construct because many methods, many researchers, myself included, have been thinking of how to optimize the construction for a decade. They are simple to define. You only need a bag of kernels. So the GFA format is a good way to represent them. They have a rich history in genome assembly. They have been used a lot. However, these graphs are typically huge because they capture every SNP variation there is in the pan-genomes or in the assembly graphs. So you end up with millions of nodes, which is a difficulty for visualization, of course, and difficulty for anything in implementation. They are confused by repetitions. Every time you have a kernel which is repeated in your genome or pan-genome, then you have a loop in the graph, and that really adds up in the hard to visualize. It's especially for biologists, it's hard to interpret individual kernels. So for instance, you can do a bacteriology analysis with these tools, and it outputs a set of kernels which are representative of differences between bacterial strains. But then biologists say, so what? What are these kernels? What do they represent in terms of biology? Potentially SNPs, potentially recombination events, but you need additional post-processing of results in order to get to biological results. So we don't capture reference coordinates, because typically when they are constructed, there is no longer the notion that some kernel comes from certain reference, even though colored Bruin graphs have been addressing this, only for the presence absence in reference, but not necessarily for abundance or reference coordinates. Although there is some recent work about that from MetaGraph. And finally, K-1 overlaps are a bit less intuitive to interpret than, for instance, variation graphs. So we'll see later which have no overlap, so it's easier to interpret. So I propose to do a small command line demo for construction of the Bruin graph. So I have my command line right here. I prepared a small repository with data. So you got those 2 genomes of E. coli that I was talking about. And I concatenated them into a single file for convenience. We can have a look at what's inside this single file by doing a clever grep command, which just shows the header and the first line of the sequence after the header of that file. So you got your first line corresponding to E. coli K12, complete genome. Then another line corresponding to the E. coli host 157, complete genome. And finally, 2 more lines which you might wonder what they are, but in fact it's 2 plasmids corresponding to the second E. coli. So these are going to be short sequences, but still they are present. So that's the data, so let's construct. A good thing about this demonstration is that I'm not using any Docker, any container, I'm just using binaries that I prepared in a folder of some of the tools that you can use to construct pan-genomes. And in fact, I made those binaries available in the GitHub repository in case you want to use them later. It's only for Linux, but still. So for the Bruin graph, we use BCAM, which is a tool like co-developed for constructing compacted Bruin graphs. And the first thing, typically, when you want to use the tool is to go to the README and check the command line. Well, I'm doing it for sure because typically I should know what the command line of my tool is, but still. You go to the README, you check a little bit what it says, and then if the README is relatively well made, you will have a usage section. It says, okay, BCAM, you give it the input, kernel size, and minimal abundance of kernels to keep. And there's an example command line here, which in fact I'm just going to copy paste and change this this. Change the kernel size to 31, and abundance minimal of 1 to keep all the kernels. Let's see if it works. Some ugly output because the terminal is too big, but it should finish hopefully soon. There we go. So, what do we get? So, there's a bunch of files I didn't remove. So, that's this one. So we get a 5 megabytes file of unities. In fact, we can output a faster file. So it's a traditional faster file with sequence and headers. The first sequence is a sequence identifier. There is the length of that sequence, the total sum of kernels inside it, mean kernel abundance inside that sequence, and then some become specific faster annotation corresponding to the edges of the domain. Using this information, it's possible to construct this faster file into a GFA file by using a script that is provided with BCAM. I give it a faster file, the output GFA file name, and the kernel length. And that should give me a GFA file, which is what I think Uni was explaining this morning. So it has segments. Here, each segment is a unity. So an ambiguous path in the domain graph consisting of kernels. And there are edges with a yellow line between that segment and other segments of the path. And let's have a look at what it looks like in practice. Hopefully it works. Okay. This graph is going to be too big to visualize. 97,000 nodes, 100,000 edges. Bandage doesn't like that at all. So the nice trick with Bandage, which is a GFA visualization tool, is that you can still cancel the layout and it will still display the graph at the end. But however, the cancellation takes a few tens of seconds. Maybe more than tens of seconds. Okay, there we go. So that's the domain graph of two equalized genomes, way too big to be visualized. But I want to try to zoom in a little bit to see why it is complex. However, the zooming in may be a bit painful. Okay, yeah, there we go. So let's get someone else on the center. So what's going on? Why is it complex? It's because you got little bubbles here. Every time you have a snip between those two strains of E. coli, that creates an event. And you see that the length of each of those two nodes is very large. It's not so big, 91 base pairs for that node, and well, 91 for the other. And so that event repeats and so on for across all the graph. So it would be better to get rid of those if we are not interested in snips. Let's do it right now. So there is a tool called GFA Tools. It is highly handy for doing GFA conversions, but also some basic simplifications of a GFA file, such as removing bubbles. It's within the ASM subcommand. It does a few assembly-related operations, but the one we'll be interested in today is bubble removal, which is that it requires a maximum radius, which is related to the length of the bubble in base pair. So I'm going to say 1,000 here, and then dash U to compact after removing the bubbles. Let's do it, and then the output will be just another name, the BU, because it's B and U on command GFA tools. It's usually very quick, and then we visualize the simplified graph. It's way smaller, 5,000 nodes. Let's see what it looks like. the light shouldn't be too long. Or is it? My computer is not happy about the heat of the case. Alright, so this is a simplification after removing all the bubbles. That's the bone graph of two equalized genomes. That's as good as we get with this k size. A lot of repetitions in E. coli at k equals 31. I'm going to switch back to the presentation now. You might wonder one thing, what if I use a different k value? And for this, I'm showing you later. So this was the graph you visualized before, without simplification. After simplifications, way less nodes. But the SNPs are now hidden. And if you increase k and simplify as well, then the graph gets even more simple, right? But still, there are some repetitions, exact repetitions of 1 to 27 base pairs inside those two genomes. What do you think is the k value after which you will have no repetitions for E. coli?
5K.
5? Or how much?
5K.
5?
Yeah.
5K, oh, 5,000, yeah. Why?
He knows. Sort of know. Isn't there that paper from Adam Philippi where they try and estimate
I couldn't hear it, so I saw.
Isn't there a paper from Adam Philippi where they...
Yes, there's a paper that shows physics particle manifold coli, so you've read it, so you know it. And the biological reason is that there is an exactly repeated gene of length roughly 6,000 base pairs in E. coli. And as soon as you have a k value longer, it's a repeated gene and everything else is not repeated, so it can't get simpler. So 127 not enough to represent in a simple way the graph. I'll just note that I tried another competitor software for constructing the graph Cuttlefish, which doesn't support large k values, so too bad. So we can do it with some effort. I tried with 300 just to see what it does. Not so interesting, it's just that 300 is not also a sufficient for jumping over sufficiently many repetitions such that the graph is complex. And finally, so we have already seen this, so GFA5 for the bone graphs looks like this. Any questions on that part before I move to part 2? Three at the same time. So
maybe it's a stupid question, but
yeah.
If when you show the SNP in the bone graph, why was it 90?
Right. I guess it should be TKM side, right?
Yes.
It wasn't. My guess is closed SNPs. Every time you have closed SNPs, the unities get bigger for the bubble. So do I still have? No. You could have investigated it, but I'm pretty sure it's closed SNPs. Or big indents.
So what's your advice, if maybe you already said this, if you want to play around like this and get larger and larger K,
yeah.
What are the best tools once you start getting to enormous K?
Oh yeah, great question. So it's still a research question. You can recompile it so that you can get large Ks. In my test, after 2000, it starts to crash and do too much more. In hi-fi genome assembly, there's the LGA software, which really specializes for construction of large K class. So I would say module 1 of LGA is your best software for that. Another question back here.
Yeah, so does removing the SNPs does it affect any sort of downstream tasks, like if you're looking at the graph and using it for some further downstream application? Does removing the SNPs affect them?
It would definitely affect your application if your application consists in finding SNPs and genomes. So this was just to visualize a graph, but if you're interested in SNPs, don't do this application interested. Good question.
Does remove SNPs just mean that it leads to the road?
Good question. So yeah. didn't say it, I should have. It detects those bubble events which are simple, but it really needs to be two paths connected to the same thing to the left and to the right. It deletes arbitrarily one of the two. It doesn't take into account the coverage. I wish it did. It would be good for assembly, but it doesn't in the current version of GFA tools. And it keeps one of the two. That's all it does. Let's get going. So VG, this might be a bit of a repetition for the talk that Eric will give tomorrow, but I'll go a bit fast, and you will hear more about VG tomorrow. My take on it is another type of tangential graph, where nodes are also sequences, and edges are no longer overlaps, but adjacencies between sequences, immediate adjacencies. You may feel free to correct me if I say anything inaccurate about this part, or any other part for the matter. That's a figure taken from, I think, the VG GitHub page by Eric Dyson. So, sorry. The first seven rows correspond to genomes decomposed into blocks, and the very last portion of the slide is the variation graph per se. So the genomes is to be pre-aligned together, and then we can construct the variation graph by just collapsing the identical blocks. My mouse isn't enough. Pros and cons. So my take on variation graphs is that there are many tools now being developed for storage, visualization, construction. So the ecosystem is very growing, especially with HPRC. The graphs are intuitive because adjacencies are without overlaps. Some tools require cyclicity, but some others can allow cycles. It seems that it's challenging to construct at scale large variation graphs. Although, of course, research has been presented this morning to alleviate this. There's still a limited toolkit for large scale. When I mean large scale, it's like human scale variation graphs, although it's of course growing. And at least at the beginning of the variation graph ecosystem, the reference was required to be set. And then Eric developed Sequish to construct variation graphs without reference by just aligning genomes in between themselves and inducing a variation graph from the alignment of the genomes without doing a progressive multiple sequence alignment, for instance. I have a quick demo for VG, but I won't use the VG software. So I will use Sequish to create a graph, and I always need to check the documentation of Sequish because it runs in two commands.
First, you need to align the sequence.
Yeah, you need to align the sequence in between themselves, and I'm doing something a bit unusual. I'm not using WF mesh just because my system has minimap2 already.
Yeah.
So I'm going to use minimap2, which seems to be allowed in the README. So I'm sure if Eric was here, he would say no, but he's not here, so I'm happy to. I'm happy to just do something better.
You will say.
Potentially, yeah, but it still works. So that takes the data. And you have to give it twice because it's pairwise alignment. Its output is just a path file, it should be quick. So that's the first step. The second step is to run Sequish. And you know what? Let me actually do something clever here. Should this work? No, of course it doesn't. No. Oh. No, it doesn't. In fact, it might take. Oh, no, actually, it doesn't take any time. Okay. now the initial thing to look at is how big is the output. It's a 20 megabyte file, so much bigger than the sum of the two equalized genomes. Let's see how many nodes does it have. It has almost 300,000 nodes. I have no hopes to visualize this in reasonable time. As many edges. So we could try the typical trick of removing SNPs. Let's see how it goes. And that's way more than usual, so let's have a look. I think it's always interesting to visualize those graphs if you have never done so. Because I had no intuition at the beginning if the graph would be very similar to the lemma, or would be entirely linear, or in between. So who's going to say like the lemma? Entirely linear, just, or in between? Most things in between. And does this look like in between to you? So, yeah, I would say it looks more like the going graph. Right? There are some strange regions which I don't fully understand why they haven't been simplified by. Oh, okay, that's because there are self loops. So you can see there's a little loop here. So the node self loops on itself for some reason, and GFX doesn't need it. So it won't simplify. And the rest, I'm assuming there's also a bubble somewhere. Well, it's strange, but there's a bubble event between those two nodes which doesn't get removed, where GFA tools for some reason. Probably because this node has an out degree more than 2. I'm assuming. Anyway, so it's just an observation. The graph is a little bit complex. Keep in mind it's only 2 equalized. And I have some more things to say, but that's all for the demo, I think. Yeah, okay, so Sequish, the raw graph, the one that I didn't try to visualize. I run it on another computer, given a few minutes, and output this. So very similar to what we are looking at here, except that instead of long nodes like this one, which is a long 80,000 K, the 80,000 base pairs long node, there are actually succession of little SNPs all across. So that's why it has lots of nodes. So I simplified it in front of you, and I also run a software that is part of the PGG toolkit called SmoothXG. I think Eric will talk about it tomorrow. It's a way to transform the graph produced by Sequish and somehow normalize it, and maybe remove some extra variations. In fact, it does have an effect. It divided my node count by 2. And in the end, the graph looks very similar to the ring graph I constructed. Does that make sense for me? Is this what you expected from the
Of course, you can get a lot of different kinds of graph in Sequish, depending on the parameters you use.
Ah, oh, that's great info. So I use default. What should I have used? What should I?
I don't know. Nobody really knows it by experimenting with it.
There is a value of k.
There's a value of k in Sequish.
Yeah.
Yes, the segment size.
Yeah.
Okay, so
There is a assumption about k value.
maybe it's inside parameters.
Yeah.
Okay, yeah, okay, there's a key value, but it's a filter apparently. Anyway, best to ask Eric, and also just this comment is enough for me at this level of introduction. So thanks for the precisions. I didn't know that, and that's what Sequish default will give you. I keep also visualizing the GFA just for the sake of it. So now you notice that the L segments have 0M, meaning that there is no overlap between nodes. In the double graph, it's a seeker string, by the way. This is zero matches. In the double graph, there was 30M, corresponding to k-1 overlaps. And some nodes can have groups. Some nodes can have just one base relation graphs. I had a question to Eric because I tried to run VG construct, thinking it would construct a variation graph, but it doesn't quite construct something that makes sense. So I will ask him later. Part 3,
I think this previous one outputs two linear paths.
Output what, sorry?
Two linear paths. And like
Two linear paths. Yeah, that would make sense. So there's one here and maybe one.
Yeah.
Do I have time for a break at some point? I'm already at that. Do you want a break? Should I continue? Yes, I hear some yes. So let's take a 5-minute break with stretches and come back at 45. That sounds good.
So what happens usually the
Alright, let's restart. I hope you had a nice break. I feel guilty making a break too long because we're on the clock here and this is part of the session. So if 5 minutes was a compromise between 0 and too long. Because there's much more coming. So there's now this mini graph, and who here knows of mini graph? And amongst who knows about it, comment how it works. Construction steps.
As it's running, or
So I'm doing for constructing a mini graph. And this is great because maybe you can help me. I don't fully understand mini graph, so I'll still present what I know. So from a high-level perspective, so these figures are only me. From a high-level perspective, this is a graph where nodes are sequences and edges are also edges. There is no overlaps. So it looks like a variation graph. The way it is constructed from a high-level perspective again is you fix a reference, which can be a graph or can be a linear genome. Here in this example, it's a linear assembly. And then you incrementally update this reference by aligning another reference to it. And through the alignment, you can induce this particular graph here, which is the mini graph for as ASM1, ASM2. And if you want further assembly inside this genome, then the assembly is aligned to the graph this time, which induces an updated graph here. The trick, of course, is how the alignment is made and how the graph is updated by this alignment. And the first part is somewhat easier to understand than the second. So it's zoomed in here. You have the reference, which at the beginning is just a single node, because it's the assembly. But here, for the sake of generality, it's a graph. The query sequence is in green. It's in green. And minimap2 algorithm is used to find seeds between the green query and the red reference, or nodes inside the red reference. So what is symbolized here? And then more stuff happens. And for this, I need to switch to another slide. So that is a description of the graph construction algorithm from the README of Minigraph, which I heavily edited to simplify where it's going. But I couldn't edit it more without a significant loss of algorithmic details. So I achieved this compromise here, which is the description of steps. So step 1 is index minimizer of the reference graph. Then for the query sequence, you find minimizer chain. It's the same as minigraph 2. between the query and the reference, and minigraph calls it linear chains. The trick is that those linear chains are then used as seeds for another chaining step in perform. So it says perform chaining again over linear chains as anchors. And then the anchors are connected using a wavefront algorithm. And there is a particularity: if it cannot connect, it still explores the original reference graph to find the shortest path between linear chains, if there is one. And the result of this additional step of chaining is what is called graph chains in the mini graph vocabulary. I don't believe they are actually the graph chain. I believe it's represented as the very last step of this graphic. And then there is a change of definitions, because you move from graph chain to primary chains, and primary term comes from mapping. It's a way to filter out some graph chains to keep only those which do not overlap significantly on the query. So it's a way to keep subchains, and this is the result of the mapping up to step 2. And for each of these primary chains, mapping quality is estimated, and base alignment is performed. Finally, this step just performs mapping. It doesn't construct any graph yet. From this set of chains, you only keep those longer than a certain threshold, and by default it's 10 kb. And then, using this filtered out mappings, there's an additional filter which corresponds to filtering out parallels of query divisions. So, if you have chains which are identical across, I never remember if it's a query or a reference, but if you have repeats, they are filtered out. Finally, from the filtered out primary chains, you keep those which are poorly aligned, which is a reference, and those which are poorly aligned are inserted inside the original graph. That's how the graph is updated. That's the best of my understanding at roughly this level of detail of mini graph. If you have complementary things to say, for instance,
I have one question. 2B isn't in the new version, because I don't know, but in the previous version it was partial order alignment.
I'm sorry, previous version?
So 2B is in the new version.
Oh.
Yeah.
Right, yes.
The previous one was partial order alignment.
Oh, maybe that's right. That's true, but the previous version of minigraph wasn't doing base level alignment.
Yeah.
And recently it is doing base level alignment. So most likely steps 2B and 2C are updated corresponding to the original minigraph application. Yeah, it's an important aspect that I think Concepto tested both. And in fact, there is base level alignment, but the overall graph structure doesn't change.
There are some complex regions that are more clear.
Ah, okay. Okay, so I guess it's good to run minigraph with base level alignment. Yeah. Right. So from this limited understanding, I could just say that minigraph is fast to construct. Again, the edges are just adjacencies, and you will see that the graphs are smaller as a result. Smaller because there's really a lot of length filtering being performed in the alignment. At this stage here, at this stage as well. And one more I didn't mention is that it doesn't even care about variations smaller than 100 base pairs. Shorter than 100 base pairs. So any snip, by definition, minigraph will not output. We can verify this later. So that's potentially a disadvantage if you're interested in snips, of course, and the construction is asymmetric. You need first preference, and then align it things to that preference. And it will not give you the same result if you permutate the input. Do we have a demo? Yeah, we have a demo. So, same thing. I need to go to the README again because I think the command line is not trivial. We don't want a map, we want to construct a graph, so that's this one. yeah, there is this command -c x GGS that needs to be run. The reason is that x is a preset and GGS means graph construction. Everything else is a mapping of sequences to graph. And L10K, I don't think I'm gonna use it, so just run it. And using, well, to equalize. And output. Actually, no, the output is not to equalize, it's a reference. The one I choose as a reference, and the other equalization. So, right, and you need to put all your genomes as a command. So far, I only have 2 genomes. So I put this one, and I will name it K12 on O1, actually not O, 157 on K12. It's the GFA.
And you can do it incremental.
Yeah, if I had a third reference, I would put it, but I don't have a third reference, so I'm going to stop it too. And if I remember correctly, it shouldn't take too long.
Because of the C command, maybe I don't know.
I'm sorry?
Maybe the C command, the new C command.
-C, oh, maybe. I remember running it with -C, it wasn't so long, it wasn't on this computer, so is my computer done? No.
Right. What
Yeah.
is the XGGS then?
Yeah. So X is a preset, GGS is the value of that preset, meaning incremental graph generation, it's a graph construction node, and -C is the same as minimap2, it's perform-based level alignment, which apparently is not recommended. It's not default, but recommended. And there's plenty of others. For instance, number of threads would have been clever for me to specify. But it's okay, it's finished in 50 seconds. Let's have a look. And when you look at that, isn't this a nice graph? I'm always happy when I see a linear reference graph. It doesn't take much. So there are some variations. They're pretty long. For instance, this one is 60 kb variant with respect to a 9 kb alternative. From visualization of this graph, it's hard to tell which one of the node is K12 and which one is O157. But thankfully, Bandage and also the new version of Bandage, all the Bandage versions have blast. So we can blast. It's blast K12. So that was K12. And I think it's a bubble here, yeah. So overall, it's going to be a rainbow. You can trace your original genome by, for instance, blasting. It's a simple way to get it. I'm sure there is potentially even annotation inside the GFA file. In RGFA, but this is RGFA, right? Because we got those elements. So I believe this is the identifier of one of my 2 equalized genomes. I would be curious to see if one of the lines has 2 identifiers and not just one. I'm assuming there is more efficient ways to do it than just inspecting by hand as I'm doing it now, but I don't have a good solution. Let's just keep this and observe this graph, and I had a few observations on it afterwards. So yeah, that's drawn differently, but Bandage doesn't have a consistent drawing value, so it's always random each time. Only 800 nodes, tiny graph, colors correspond to the blast of K12 as you just saw. There is one thing that is glaring in this graph. What is it?
It's missing the inversion.
Yes. Where is the inversion? There should be an edge from, I don't know, here to here. I didn't find it. So, yeah, good question.
No, nothing.
So, is it somewhere I missed it? I don't think.
Didn't your description of the process say that it chucks out the aligning bits?
Yeah.
So could 1/2 the inversion have been chucked out because it looks like a...
That could very well be it. So that's an open question to hang. Are inversions just removed from mini graphs by design? But I think it's good to keep that in mind when you visualize pendulum graphs. You see what you expect to see.
It's going to be very hard for him to... In that text you have from the read, I think didn't it say it wanted to get rid of duplication, big duplications?
Yes, parallel distributions.
So, I mean, you can't really tell the difference between an inversion and a parallel very easily.
You should, because the parallel is within the sequence. It's not an ortho.
I mean,
so
you're right.
So
I can believe that getting rid of one axis.
sorry?
I can believe that getting rid of one axis, so that's definitely a really good. I'm just guessing.
It's very likely. I don't have a good answer at this point. Let's just leave it at that. No insertion. That's what you have. But it's still, apart from that, it's quite great as a workable pendulum graph, right? It finds positive variations. So it looks like this, long nodes annotated using specific RGFA annotations, which are S something flags in GFA. Okay, this is reference name, this is coordinate, and this is the rank, which I never remember what rank is. Is this some sort of genotype?
The rank is basically zero is the original reference, and then when you get a node from later sequence, it gets increased. It's increased the rank.
Great. Okay, thanks. As you can tell, I'm not using mini graphs so links. So same, no overlap. Annotation of 1 and 2, which I suppose correspond to the length of the input and output segments, I would say, and ranks. Right, any questions on minigraph? If not, I move to the last example. Final time.
Yes, sir. Maybe I lost it, but what's the method used to align the new query to the graph?
Minimap 2 like seeding and chaining.
Oh yeah.
It was, it's essentially 2A.
Okay.
But there's an additional chaining performance. There is a specificity of minimap. Okay, can anyone guess what is part 4? It's not even told to say.
Pandora.
Sorry?
Pandora. Is it
Pandora? No, it's not Pandora. Could have. Maybe it's Pandora. So part 4 is MTBG. It's one of my projects, so I had to mention. And Zan already talked about some of it in his talk. So thanks for the kind words. So MTBG is a variation of the volume graphs where nodes are special k-ners, so I call them k-minus, k consecutive minimizers. And edges are k-1 overlaps, but not over bases, but instead of minimizers as well. So what's going on? You take a reference genome, and could also be reads. You extract minimizers from this genome. And the way the minimizers are extracted, I could talk for a while about it, but just think of it as you have a predefined set of minimizers that you expect to find. And as soon as you find them, you will keep track of them in the order that appear in the genome. So look at the genome from left to right, you find AC, but the next minimizer you expect to find is AG and AC and T and AC. And from this linear order from left to right, you extract kernels of consecutive minimizers. Considering that the minimizer is now a letter of your alphabet. And once you do this kernel extraction, here k is equal to 3. The minimizer alphabet. And that's all there is to minimize space decoupling graphs. So it's useful for representing low resolution graphs for a large number of genomes because it's quite efficient. For instance, it's possible to visualize a whole human genome. I think this one was created from CHM13 hi-fi reads. It's not the best assembly you can get from this dataset. For instance, T2T much better. But you can obtain a decent looking assembly from those reads by just performing an MDVG and some level certifications. And there are some advantages and disadvantages. One is that it's super efficient to construct, and it was applied to the representation of what I think is the largest genome that exists, 600,000 bacterial genomes, but that compiled painfully with this group. The graphs are really easy to visualize by virtue of the fact that we don't have so many nodes. They produce smaller graphs than any of them. However, you lose a lot of resolution. For instance, SNPs are completely discarded except when they fall exactly at the position of minimizers, which might happen, but not so often. For large graph, for instance, for this collection, the resolution, which means the span in terms of the base pairs of a node, is in the order of 10,000 to 100,000 base pairs. So each node of the graph represents 100,000 base pairs. The toolkit is very limited because we are the only group who really work on it so far. But, so this is some visualizations of human MDGs. On the left, it's from the assembly, and on the right, it's from the reads. Notice that, well, it's more contiguous. You start from assembly, there's no coverage gaps. It's overall more simplified. Because, for instance, this individual is deployed, or almost deployed. It's more fair to visualize a deployed human genome. So this is an NTPG of the sample HG002 assembled. So I took 2 haplotypes and gave it to NTPG. And you can see here that every time there's a color change, it means that it's a different node. Actually, do I have some inversion? Yeah, I have some inversion. There's a lot of bubbles here. Each node corresponds to, I think it was 100,000 base pairs. And those are large variations in units, due to the fact that it's a deployed individual. And this is a genome graph of 5 human haplotypes for human. It was quite efficient to construct, so a few minutes per haplotype and 6 KB per haplotype, just because the software now is limited by faster parsing. So the method is efficient. And a quick demo on Nikolai, that will be the last one. So we had a tool called Rust MDBG. Which might not have a good readme. Yeah, well, it's sufficient. I guess we have a quick start readme to explain how to run it. It has a few parameters, the k value, of course. The density is roughly how often you expect to see a minimizer in a sequence. So a low value means that there's a lot of space between minimizers being sampled. For instance, the density of 1 is at every position of your genome you have a minimizer sample. density of 0.5, only half of the positions. So 0.008 means 1 over 10,000 positions will be sampled by a minimizer on average. So that's how you get a drastic reduction in graph size. L is the minimizer length, minimal abundance of k-mers, and that's about it. Take K2 a density of 0.00, let's go 0.001, so that's 1 over 10,000, and a minimum abundance of 1. Should work. Oh no, I need to put reference as well. That's it, you only have 4 nodes. That's not right. Oh no, let's only have 4 bits, sorry. 1,000 nodes. I don't want to visualize the GFA, I think I just have a GFA. Yeah, it's this one. I had a previous execution, but it's this one. Let's write 1,000 nodes. And we get a very different graph from big graph. Why is that? Two genomes. That's right. I realized I didn't make unities out of this graph, but let's do it right now. And in order to do this, I need to convert it to base space. I will convert it to base space at the same time. So for this, I have a tool called toBaseSpace, which takes as input the sequences and the graph. Alright, so that's the first time I get a bug on this. So do you have a problem? Oh yeah, okay, I need to create unities first. Right, so the input of toBaseSpace requires unities. So now we got graph, which is it's a bit of a mouthful to pronounce, but it has all the parameters inside the name. It's completed in base space.
Ryan,
Yeah.
is it base space unities or k-min unities?
Okay, so I should be more precise. So Rust MDBG software that I just ran outputs k-min non-unities. So the raw green graph not even compacted into unities, k-mins. Then I ran GFA tools to convert my k-miners into k-min unities. And then I run toBaseSpace, which takes as input the k-min unities, a binary file produced by Rust MDBG that contains the sequences of each k-min. And then it reinjects those sequences inside the k-min unities to give me a graph that is called complete. It is now base level unities MDBG compacted. So yes, that's a lot of things going on, but 3 steps. Let me just make sure that it looks like what we just observed before, except now it's compacted. And oh, that's not really great. What is this one? What did I just generate? Ah, I generated an empty graph. Yeah, that's not great. Do we have a backup here? Ah, yeah, this is probably better. Yeah, that's like a graph. Yeah, I wasn't opening the right file. Okay, so that's a base space with MDBG, 2 genomes, that are sufficiently different always get separated in k-minimum space due to the SNPs being captured by minimizers. And those two equalizers are sufficiently different. Usually the threshold is around 1%. Let's blast and confirm this. So I blast K12 and we should get one of the two parts of each bubble corresponding to K12 and the other one not. We don't. Why? Any idea?
It's just, no, there's a, so they can map to both, perhaps if there's a percentage of identity that you can.
Absolutely. Let's go with 99.9% identity in blast. We blast it.
I'm sort of surprised the classes are fast.
Me too. I don't know. Alright, so you got your K12 path and you see it's mostly separated except for some region here. You've got the 34 KB region common between K12 and sufficiently common minimizer space between K12 and O157, but it becomes just one node. Sometimes it doesn't happen and we just add these connections. So yeah, that's all I wanted to show for these two genomes. What else do I have? That was with different set of parameters with 10 times bigger density, bigger graph, but still relatively small. And
Sorry, if you go back, how can we, if we just have two genomes, one more, please. How can we?
one more.
Sorry.
This is the only slide.
One back, I think, the one where you were, oh, sorry, on bandage. Sorry, it was a bandage, yeah. So, if we have two genomes, how can we have full connections between two segments there?
Oh, you see 4 connections?
No, I mean,
Yeah.
here, so,
This, right?
yeah, exactly. How can we get that from just two genomes?
Right. Clearly, 2 of those paths out of the 4 are not correct, don't correspond to a genome. So what could happen is that you have a similarity in minimizer space between the extremities of those 2 mutate. And the way it has been compacted is that instead of creating a single node, it created 2 nodes.
So you mean you have map now, if we now map the other one, probably should take the 2 on those?
Oh, I surely hope so. Let's do it.
Okay.
I surely hope so. It should be the 2 top. Yeah, it's the 2 top. So clearly, this one's correct. This one's correct, but this jump from here to here
Yeah, that's what I.
is not biological.
Isn't that sort of the difference between a square graph where you store all the edges effectively between compatible pairs and only the edges that you see in the data? They're 2 different representations.
Absolutely. So yeah, so it's a k minus 1. It's that overlap in minimizer space between those 2 parts.
I'll do this to me, sorry.
Yeah, so it's exactly the same.
I was thinking of, it was an overlap, so.
Yeah. You have to fit in the void space for this.
Yeah, I see that.
A good question though. It still puzzles me that you have a k minus 1 overlap, but not an exact k minus repetition. Anyway, that's what's happening. Any other question? I have some explanations left of what's going on. So if you decrease the density, you increase the probability to see common elements between one integers. For instance, this little node here, this little node here as well, and so here. MDBG by default looks like this. If you just run the software last MDBG, it doesn't output any sequence. Then 2 base space will output the base space sequences. I provide an estimation of what the overlap length is between nodes, but it's really an estimation based on minimizer distances and an accurate one. Same for the length of the nodes. It's given in base space, but it's also an estimation based on minimizers. Oh yeah, I have part 5. Maybe not so much time. I just wanted to show and discuss one thing, which is to discover things from pendulum graphs alone. Not by mapping weeks to the pendulum graph, but just constructing the graph. Can you get some information from it? Maybe structural lines. And the case I wanted to make is that it's maybe possible to discover something interesting from this you have here, which I've constructed but not analyzed fully. It's MDBG of all the non-equal genomes. It looks like that. It's very surprising to me, but it should look like this. Because equal genomes have a large open genome, so they have lots of genes. And it's very surprising that you can almost trace the consensus sequence inside this graph, because in fact there is not a consensus sequence of E. coli. For the genome of E. coli, there is not one sequence that symbolizes all of them. Somehow the graph seems to imply there is some sort of backbone. So I will skip what's next, but briefly, I did some preliminary analysis on this graph by zooming in, looking at the size of the nodes corresponding to how many genomes are represented by these nodes. So the thick one is maybe some sort of backbone, nodes seen in many different E. coli. The thin one corresponds to chimimers seen in very few E. coli. And then we can notice that it's a mess. And zooming in particular parts, we can see that there are some interesting connections between backbone nodes, which putatively correspond to, by definition, structural variations between multiple E. coli genes. So the information seems to be there, but there is no automatic way to detect those events from graphs as far as I can tell. So that's pretty much where I stopped the analysis. Apart from confirming that for this particular edge here, it really corresponds to potentially a genetic exchange between K12 and some other strain of E. coli. Because what's on the left of it maps well to K12, what's on the right maps also well to K12. However, the sequence associated to that edge has a split hit between two different regions of K12. So if it's a split hit, it means that something has been inserted in the middle. Okay, I won't go much more further. So today, in this hour and a half, we've constructed and visualized a good amount of pendulum graphs with DBG, Sequish, mini-graph, and DBG on 2 E. coli genomes. So my conclusion is that it's all GFA, which really simplifies visualization a lot. Using Bandage. All the tools were distributed as single binaries, which simplify execution a lot. However, for some of these tools, binaries were not distributed by the GitHub repository, but they were present on Colab. So you can, in fact, Google the name of the software Colab and then extract the binary without even having to install the software. Large case, still in research for efficient construction. VG has a growing toolset, as you see. Minigraph, I really like it because it's so fast, so small graphs, and apart from this missing inversion thing, it seems to produce what you would expect from a linear pendulum graph of your genome. And DBG is also fast, also produces small graphs, but it's highly sensitive to small variations. That's why it separates genomes which are sufficiently different, which was the case in this example, it doesn't separate human genomes that much. A brief, just one slide to explain that at Pasteur we are part of Alpaca, and what I showed today on 2 E. coli, Francesco is actually doing this type of things on 100 human genomes. And you can imagine it's much harder to construct, analyze and understand differences between those implementations. We're working on a paper to talk about all this. Alright, that was my last slide, so thank you.